going to be talking about the cohort study that we currently run, ACORN, and its upcoming developments into a study called ACORN Plus. And I know a number of people here are involved in ACORN already, so I'll be able to tell you a little bit about why we're doing what we're doing and some of the work, some of the research work that's going on with the samples and data that you've been providing. So the key point, the key motivation really for the work of these cohort studies is that we want to make ALS and FTD preventable diseases. But there are a lot of steps along the way that we need to achieve before we can make that into a reality. So we need to have a better understanding of who is at the highest risk of developing ALS. We know that people who carry a genetic predisposition to the disease, a proportion of those people will never develop a problem, will live a normal lifespan, won't develop ALS or FTD. And so understanding within the group of people who have a genetic predisposition who actually is going to go on to get on the disease is really important. So we know who to target treatments towards. We also need to understand when that is going to happen and what are the early changes going on that sort of predict that someone is moving from that compensated phase that Martin talked about into the disease phase, so we know when is the optimal time to be treating people, when treatments become available, or using tofersen, for example, in SOD1. And a lot of the work we do, again, primarily focusing on that compensated period, is to try and understand what is happening that allows somebody to tolerate having that abnormal gene for decades of life before this catastrophic change happens with a progressive illness. And through that, we will understand some things about risk, what influences the risk that somebody has that we might be able to intervene in. If we can understand the mechanisms that are going on inside the body, we might be able to augment those as a means to bring about prevention. This is difficult work. This is a difficult task to address. So this, we're dealing with what is quite a rare disease. So the genetic forms of MND more so. So there are around 12 to 1,300 new cases of ALS diagnosed each year in the UK. Of those, around 10 to 15% will be associated with a single genetic change which is causing the disease. And when we're looking at the compensated period, as well as looking at the people with genetic forms of ALS, we are very interested in looking at people who we know are at risk. And from each of those newly diagnosed people with ALS, we will have typically one or two people who our family members know they're at 50% risk of carrying the same gene, who we can ask to be involved in the research. So the numbers there aren't small, but actually, really, we need to be able to access as many of those people as possible to make this work. In that early compensated stage, there is no outward evidence that there's a problem. So we expect the differences that we're looking for in the compensated period to be very small. And as a consequence, we need to be studying a lot of people in great detail to be able to detect those changes. We need to monitor people or observe people, study over a period of time to understand how things change with time. That's of really key importance to this and is a very sort of central part of the way that these studies have been designed. And because of these challenges, we recognize this needs to be done on at least a national scale and ideally really an international scale, making sure that all of the data and samples that we collect is usable by researchers working in the UK, but also researchers working overseas, so that we can really take advantage of the scale of international work. These genetic forms of the disease, although they vary, their distribution varies geographically, they're present in all corners of the world. So we're taking a couple of different approaches to addressing this, and you're going to hear about both of those. So one of those is we're taking advantage of large population-based cohorts that have been established. So here I've singled out UK Biobank which is a UK study, All of Us, which is in the United States. So these are studies where 500,000 people from across the country in middle age have been recruited. They go to an assessment centre, give blood samples, provide information about their health and lifestyle. Over time, some of those people have gone on to develop ALS. Some of those people have genetic risk of the disease. Yeah? And so we can study what's happened to those people that started with a genetic risk over time to try and understand some of the questions that we want to answer. The problem with these studies, though, is that they're not really focused on ALS or focused on neurodegenerative disease more generally. They're a lot more about general health. So they don't study nervous system health in quite the detail that we need. They're also not longitudinal in the way that we need them to be. So, i.e., the sampling, for example, is mostly done at a single time point. And that means it's harder to understand. If we see changes in the protein makeup of the blood, it makes it harder to see how that changes over time in the people who are at risk, so that we can really understand how the sort of time course of changes. So that is the motivation for studies like ACORN, which we currently run. So with ACORN, we can do much more detailed study of people. We can understand a lot more. We can do it in a much more detailed way over time as well. And it enables us to ask specific questions that we can't ask in these big data sets. And actually the way that we approach this is, we ask questions, we will start with a question in the big biobank data sets and then see whether we can answer it there and then also in our more detailed studies, or vice versa. So in terms of ACORN, there are sort of two elements to it. One is it acts as a register of people with or at risk of carrying the C9orf72 repeat expansion. So that is people living with C9orf72 motor neuron disease or frontotemporal dementia and their at-risk relatives. So typically children, siblings, in some cases parents. And the purpose of the register is so that if there are research studies that come up, we can share information about those with people so that they can take part, just to accelerate research for all forms of research that are going on in this area across the UK. The other element is this detailed national cohort study. And some of the key features of the study, which if you're involved with it, you will be aware of, are that the genetic testing within it is done in a blinded way. So people come and take part in the study without knowing their own genetic status. Some people will know their genetic status having been tested through sort of clinical pathways in the NHS, but most people will not know their genetic status, and that's of key importance, so that people can take part without knowing and with no risk that they will find out their own genetic status as part of the research. So the genetic samples are taken, the testing is done separately from the study and then matched up with anonymized data later, so that there's no risk that an individual in the study or a researcher involved in the study will know an individual's genetic result. There's no risk of us inadvertently disclosing what is life-changing information. For the cohort study, we have two different visit types. People can come to Oxford for a detailed study visit, which I'll talk a little bit more about, and we also do remote visits so that we can try and capture as many people as widely geographically as possible, recognizing that people living in remote areas or people living with disability cannot travel for the more detailed study. And thinking about the fact that this needs to really be a very large international effort, what we've done is we've made sure that, as far as possible, elements of the way that the study is conducted aligns with other research studies in the same area happening in the UK, like GenFi, some of you may be familiar with, but also international studies elsewhere overseas, so that we can use the anonymized data together to improve the power of what we do. So study visits in ACORN, many of you will be familiar with this, involves coming into Oxford, detailed questions about lifestyle, medical history, family history, cognitive tests, physical examination, and then we do we take off blood samples, and if willing, people will have a lumbar puncture as well. Now you might ask why we do a lumbar puncture, and not everybody wants to have a lumbar puncture, and that's okay. What I would say is that the reason we do lumbar punctures is because we access cerebrospinal fluid. It's the fluid that bathes the brain and the spinal cord. It's as close as we can get to the cells affected by the disease without actually taking a piece of the cells, which would, as you can imagine, be problematic. And we do this, if people are willing, each time they come for a visit. The visits happen every 1 to 2 years. We also do some MRI scanning of the brain, which tells us about the structure of the brain. We do MEG scanning, which Martin mentioned, sitting in this sort of oversized hairdryer, which measures millisecond by millisecond brain activity and how the areas of the brain are connected to each other. So far, we have 124 people enrolled in the study, which kicked off in December 2023. Of those, the majority are people who know they're at risk of carrying C9orf72, with a smaller number of people who have either ALS or FTD, and people who are sort of interested in contributing, so-called controls. We've got participants from England, Scotland, and Wales. We don't yet have anyone from Northern Ireland, but the geography is an important consideration that we're going to go on and try and address. There's also initiated in Australia recently with a collaborator, Thanuja Dharmadasa in Melbourne. They have initiated a sort of sister study based on the same study design in Australia, again, making sure that we can compare and use all of the data that we develop together. So some of the gaps that we are conscious of in ACORN that we're looking to now address are that it only includes one gene. And as Kevin and Martin have already pointed out, there are a number of genes that cause ALS. And studying these different genes may reveal things that tell us about the shared pathways that lead to ALS, and also the ways that these genes differ in why they lead to increased risk. As I mentioned, the geography is an issue. It's a bit skewed. We are skewed towards the south just because we're in the south. And this is particularly the case for the in-person visits. So we've got better coverage with our remote visits, but we do want as many of the detailed visits to happen as possible. And we really want to be able to capture everybody across the country who is willing, so it needs to be bigger than it is. So I can be very pleased to tell you that the MND Association, we are sort of in late stages of discussion with the MND Association, who've agreed to financially support us to expand ACORN into ACORN Plus. I'm sorry for the lack of a really great new acronym, but we like ACORN, so we thought we'd just make it bigger. This will involve studying people with additional genetic forms, so particularly focusing on those big 4 genes that Kevin mentioned, as well as others. So we really want to involve anybody where we think that the gene is causing ALS in their family. The target is that we will recruit 300 people to ACORN Plus over the next 4 years. We're incorporating additional sites, and actually there are some people from the additional sites here today talking to you. And initially that's going to be sites at University College London and in Sheffield, which are 2 centres that do research in frontotemporal dementia and ALS already. So we've got Rhiannon Convery, who's going to be contributing to the study, who's talking to you later, alongside John Rohrer and Pietro Fratta from UCL, and from Sheffield, Alistair McNeil, who's here today, and Jonathan Cooper-Nog. As this develops, we want to incorporate more sites as well to help us access some of the more remote geography. So in the southwest, Belfast, Wales, where we have particularly, we have a bit of a geographical gap. Another important element of ACORN Plus is that we want to improve the way that we disseminate information about the research that we're doing to people who have an interest in it. So some of that is going to involve development of resources and development of a website that's around the study. That's partly to encourage people to take part and build the study, but a big focus is going to be about how we share information about genetic MND and genetic risk, and also share information about the kind of research that's going on, other studies people can take part in, but importantly, the results of the research that we do and how it is relevant to people. And this is something where we're going to be wanting help from you to help guide us in terms of how we share that information. And Daniel, who many of you will know as our project manager of Acorn, will be asking for some input on that in due course. So we expect this really Acorn Plus to be kicking off over the next year. Next, I thought I would just really touch on some of the research, the sort of scientific work that we're doing with the samples and data that we've acquired in Acorn. So the first of these is to say that we are conscious that in SOD1, we're already in this position where we're now doing a preventative trial of targeted gene therapy. When we have gene targeting therapies for the other genetic forms, we want to be ready to be able to roll those out, as soon as possible. So we're doing a study called Monitor C9. I'm just going to call out Corey Holdham and Isabella Ramanath, who are both at the back there. You're welcome to chat to them later, who are helping to run this study, which is about can we remotely monitor people who are at risk of MND to look for early signals. So some of that is with finger-prick blood tests that people can package up and post into us. Some of it is wearing wrist-worn activity monitors, a bit like a sort of Fitbit, but it doesn't tell you anything, it's just blank, that people wear so that we can see what are their activity levels like and things like this, as well as remotely measuring brain function using an iPad-based set of cognitive tests. We're also doing work using the samples that people have given, looking at that compensated periods to try and see what is changing there, what differs in people who carry one of these genes compared to people who don't, that might give us clues about how the nervous system is coping with that genetic change. So this is some work that we've done in Oxford. Lizzie Dellar, who's one of our postdoctoral researchers, this was a collaboration with UCL as well. We measured the levels of thousands of proteins within the cerebrospinal fluid samples that people had given. And we could see that there were levels of a protein here called UCHL1. Actually, unfortunately, the rest of the proteins haven't really projected, so it looks like we've just got one dot on the graph. But this protein called UCHL1, we found that levels were increased of that protein in cerebrospinal fluid of people who carry C9orf72 at actually young ages, and the levels appeared to increase in ages more than they did in people who didn't carry the gene. There's a lot more work to do to understand what this means, but this protein is something that may well be involved in the compensatory mechanisms. It may be a marker to tell us that the nerves within the nervous system are somewhat unhappy. We're not sure about that. We're now taking this protein and studying it in some disease models to really try and understand where is this signal coming from, what is it telling us, is it something that we might be able to target. You're going to hear a bit from Andrew Douglas later, who's a geneticist who's doing some work with us. And he's very interested in understanding why the risk of ALS and frontotemporal dementia appears to differ between different families with C9orf72. So even though it's the same gene, why is the risk different in different families? Which may hint at other genetic problems. You're going to hear more about that later. This is using the family history data that we've collected through study visits in Acorn. We're also doing work with the DNA samples that we have as well. So this is work that's done by Beatrix Cardus, who those of you who've been for study visits may well have met, and who is also at the back, or you may have seen her on the way in. This is looking at how the size of the expansion in C9orf72 influences risk of MND and frontotemporal dementia. So this is a picture of the C9orf72 gene. I would say this is how long it is. This is not how long it is. It's about 0.002 of a millimetre long. But this is it sort of magnified, okay? That little bar there is the repeat in a normal, in a normal gene with no expansion. So normally there will be 2 repeats of this 6-letter sequence, GGGGCC. Yeah, so 2 repeats. Some people will have 5 or 8. Don't know why it's those numbers. Okay, in somebody with a C9orf72 hexanucleotide repeat expansion causing ALS or frontotemporal dementia, typically there will be something in the region of, well, it's usually hundreds to thousands of that repeat. So this is what it would look like if there are 1,000 repeats there. So you can see that there's this, it's quite big in relation to the size of the gene overall. And you can imagine that when the body is trying to read the letters of that code, that having that there could interfere with that. Now, there are a number of neurological diseases that are caused by abnormal repeating sections of DNA in different genes. Probably the most famous example is Huntington's disease, where you have, it's a different gene, it's a different set of letters, and in Huntington's disease and in other repeat-related neurological disorders, the number of repeats relates to the severity of the disease and how early the disease comes on. And in C9orf72, there isn't really consensus about whether that's relevant or not. So one of the things that Beatrix is doing is using some very advanced techniques to measure individual strands of DNA from cells to read every single letter across the repeat, to tell us how long is that repeat. Are there areas where the repeat has got little interruptions in it, and how that relates to the risk of ALS and FTD, and how it relates to age. One of the other aspects that Beatrix is looking at is she's looking at some tags that the body sticks to DNA as well. So these tags are called methyl tags or methylation. So it's a sort of little chemical addition that the body puts on to bits of DNA where it wants to turn a gene off. And one of the observations is that that abnormal repeat has extra tagging on it. And one of the things that Beatrix is doing, again, is looking at this tagging and seeing, does that relate to if somebody has disease or is healthy? Does it change with age? What's the relevance? Okay, so that was really all of the aspects that I wanted to touch on. So I was just going to summarize what we talked about. So ACORN and ACORN Plus really is a study and register of people with or at risk of genetic forms of ALS. We're planning to expand that over the next year to multiple sites and to incorporate more genes. We're doing ongoing research to try and understand who is at highest risk, when is that risk, and can we predict when somebody's going to run into problems, as well as what alters that risk in a way that we might manipulate to help with prevention. Got a QR code there if you're interested in finding out more. It just links to the study website, which we're trying to improve, or the email address for the study if you're interested in learning more or contributing. And finally, just to say thank you to everyone involved and our funders.